Hello everyone, myself Palash Manoj Thakre, and today we are going to learn about a very interesting topic of transcription, that is RNA polymerase in prokaryote. Dear friends, before moving towards our topic, we must have to know about what exactly the basic features of RNA polymerase enzymes. There are different forms of RNA polymerase enzyme in a living organism, but in the prokaryote, only single type of RNA polymerase enzyme performing same reaction for all the different types of RNA. We can say that although there are different types of RNA polymerase enzymes are there, but they performing same type of reaction. It is identified that the shape of the RNA polymerase is non-spherical. If we talk about the size of RNA polymerase, then assume that the eukaryotic DNA containing a promoter region. This promoter region is made by 60 base pair sequences. Out of 60 base pair sequences, the RNA polymerase bind only on the 16 base pairs of promoter region. It requires magnesium ion for performing its activity. Without magnesium ion, the enzyme is not getting activated. The rate of transcription in prokaryote is 40 nucleotide per second. It means that RNA polymerase enzyme polymerizes 40 nucleotide per second. The rate of transcription will be effective at 37 degrees Celsius. Let's move towards the structure of RNA polymerase. The RNA polymerase, it is called as multi-subunit complex. It is built up by 5 different types of subunit, and each subunit is itself a protein which has a specific function in the transcription process. The RNA polymerase, it is called as core enzyme. It is an enzyme which is made by 5 different types of subunit. These main 5 subunits are 2 alpha subunit, 1 beta subunit, beta dash subunit, and omega subunit. These 5 subunits are destined to form a particular core enzyme complex called as alpha 2 beta beta dash omega complex, which is actual real transcription enzyme called as RNA polymerase. The RNA polymerase does not recognize the promoter region on the DNA directly. Now the main question is here, how the transcription will get initiated? Yes, for the initiation of transcription, the core enzyme complex required a particular factor called as initiation factor, and that initiation factor is sigma factor, which is useful for the initiation of the transcription process. The sigma factor bind to the core enzyme and the enzyme will become holoenzyme, and the complex will become alpha 2 beta beta dash omega sigma complex. The sigma factor is required only for transcription initiation. The sigma factor is released from the holoenzyme complex, leaving the core enzyme after transcription initiation. For making a proper sense of RNA polymerase structure, just observe and look out the following figure. As we already discussed that the RNA polymerase structure is made by 5 different types of subunit. 1 beta dash subunit, 1 beta subunit, 1 pair of alpha subunit, and a sigma factor which will make this core enzyme to holoenzyme. Let's discuss the different function of different subunit. The beta dash subunit, it is the largest subunit in the RNA polymerase enzyme. It is encoded by RPOC gene. It means that when the RPOC gene getting expressed, then it will form a particular product, and that product will be protein, that is beta dash subunit. The beta dash subunit and beta subunit together form a particular center called as active center, which is called as active site of RNA polymerase. Hence, beta-subunit it is the also part of active center. It contains the determinant for non-specific interaction with DNA and nascent RNA. The heparin has shown to bind to the beta-subunit. It means that the heparin will compete with the DNA and inhibit the transcription process. The beta subunit it is the second largest subunit of RNA polymerase enzyme. It is encoded by RPOB gene. It means that when the RPOB gene getting expressed, then it will form a particular product, that is protein. It is also called as beta subunit. As we already discussed that the beta dash subunit and beta subunit together form a particular site called as active site, which is the active site of RNA polymerase enzyme where the reaction takes place. We also say that the rest of determinant for non-specific interaction of DNA and nascent RNA is done by beta subunit. Here the meaning of determinant is it can be determined or it can be isolate or evaluate whether the particular sequence is made by a proper specific nucleotide or not. It is claimed that the rifampicin and streptolidins these are the antibiotic which can bind to the beta subunit and inhibit the transcription process. The RNA polymerase structure containing one pair of alpha subunit. These are alpha 1 and alpha 2. Each alpha subunit containing two domains, that is alpha NTD and alpha CTD. The alpha NTD containing N-terminal domain which determine the assembly of core RNA polymerase. Alpha CTD contains C-terminal domain which determines and evaluate the interaction of RNA polymerase enzyme on the promoter site of the DNA. The omega subunit it is the smallest subunit of RNA polymerase. The main function of omega subunit is it induces the polymerase to stabilize its structure. As we already discussed, the sigma subunit it is called as the initiation factor, which initiate the transcription process, which will help RNA polymerase to bind at the promoter region specifically. The sigma factor is released from holoenzyme complex after the initiation of transcription. Thank you very much guys for watching my video. In the new lecture, we will discuss about what exactly the transcription process in detail. I hope this video will very much beneficial for you all. Thank you very much again.